Hey Shefali, how are you doing?
Hi Kushal, I'm all right.
Okay, what happened? Is your ML project not working well? If I remember correctly, you were trying to predict patient survival using gene expression data, right? Could you not get the desired accuracy?
Yeah, we collected as much data as we could from Polyomics Atlas. We've tried as many different possible models as we could, and we've tried to optimize these models. But the accuracy seems to have hit an upper limit of around 70%, and it's quite frustrating, and we don't know what to do next.
Yeah, I can understand. As you know very well, biomolecular data like gene expression can be very noisy and also has high dimensionality. So, for example, the data that you yourself have, it has only a few hundred patient samples, but 20,000 gene measurements in each sample. Maybe that's the reason for low accuracy.
I'm sure that may be part of the reason, but we've tried to account for it through different dimensionality reduction methods. And I'm starting to suspect there's more to the problem. And one of the reasons may be that survival is a macroscopic property, and the data that we are using, gene expression, is a microscopic property of a biological system. So I'm beginning to wonder if this data even has enough information available within it to be able to predict a macroscopic property like survival with high accuracy.
Yeah, I think you are right. Biological systems actually are multi-scale, even at the microscopic level, with so many different interactions happening between genes, proteins, metabolites, and there's so many different modifications.
Yeah, and this is also probably why multi-omics data analysis is becoming so popular these days, because just one type of omics data is not probably sufficient for many problems to make clinically significant or relevant predictions with high accuracy.
Yeah, true, very true. I have actually seen some papers on ML analysis using multi-omics data. But the problem there is that proper integration and normalization of these vastly varied data sets that can be highly non-trivial. Of course, there are some statistical techniques which can help in addressing these problems, but these are mostly domain agnostic techniques. But I strongly feel that for biological data, especially, incorporating domain specific knowledge is going to be very crucial for making predictions with high accuracy.
But wouldn't this knowledge infusion destroy the whole purpose of doing machine learning analysis? Like, isn't it better to take multi-omics data and let the model figure out the patterns in the data instead of biasing the model by adding prior biological knowledge?
That's an interesting point. But why exactly do you think knowledge infusion will bias the ML model?
Well, so as I see it, the purpose of using machine learning with biomolecular data is to help discover new things about that data that were previously unknown. And it seems in conflict with the idea of knowledge infusion. So if we already have relevant biological knowledge about the problem, then why use machine learning at all?
Okay, I see your point. So that is just because biology is very different from physics. Of course, both of them fall under the umbrella of natural sciences, but conceptually, they are actually very, very different from each other.
What do you mean?
So, for example, let's take physics. As you know very well, all the knowledge in physics is encoded in the form of well-defined mathematical equations, which are usually sufficient to describe the system being studied. We can even make very good predictions about the experiments which are being performed using the already available theoretical equations. But in biology, such equations exist for a very few cases. And as you know, most of biological knowledge is actually determined experimentally. For example, let's take the codon AUC. We know that it always codes for the amino acid isoleucine, but do we really have an equation that can tell us how many of these amino acids will be produced in a cell in a given time interval? So if we have this information available to us from biological experiments, we could infuse them into our data, which is called knowledge integration, and that can help in improving the performance. This is just like filling some missing data in your data tables.
That's very interesting. So, if I understand correctly, what you're saying is that if we add prior biological knowledge in addition to the molecular data and give both together to our machine learning models, it can potentially improve predictions by giving the model more information to act on and maybe making them more robust to noise. And that's very common in biomolecular data. So I think how much it improves predictions is still an open question for us, but I think it's definitely an interesting method for us to try.
Yeah, actually, Shefali, now that I think about it, you have actually been using knowledge infusion already. To begin with, for example, you had 50,000 genes, but you selected 20,000 of them for your analysis based on your knowledge of that particular disease, because you knew that those 20,000 genes are the ones which are relevant for the problem you are studying. So almost every ML project with biomolecular data, I would say, infuses biological knowledge in one way or the other. But the problem is that it is not done in a very systematic way. But I can bet that it is almost always done in any ML project with biomolecular data.
You know what? I think you're right. Some prior knowledge definitely goes in almost every project we do. And it's not just in the choice of features, in this case, for example, us choosing certain genes over others, but also in how we choose data sets and how we choose samples to include. So that's very interesting.
Awesome. I think it can be a game changer if we can actually infuse and combine this prior biological knowledge with biomolecular data in machine learning projects in a more systematic way. We have so much biological knowledge which is available from decades of experiments, which are stored as pathways, interaction networks, and various kinds of ontologies.
Yeah, that's certainly a very interesting direction. So I think one of the simplest ways for us to apply this to our project would be to take pathways from, say, gene ontology and use pathway expression as features into our model instead of just individual genes as features, like we've been doing so far.
Oh, great. That would lead to a substantial reduction in the number of input features as well. But wait, just to make sure that I understand your idea correctly, let me take a simple example. So let's say for the survival of a cell under certain disease condition, we know, let's say from biological knowledge, that 15 different pathways are required to be functioning properly. And let's say each of these pathways has, let's say, 200 different genes. So in total, we have 15 times 200, which is 3,000 different genes, which would be your initial input features to your ML model. But now let's say if we infuse biological knowledge, so instead of 3,000 genes coming into the model separately, we can divide them into 15 different groups. Each group represents a particular pathway, and then we take a mean of these 200 genes in each group, and that goes as features into our ML model. So now instead of 3,000 genes, we have only 15 mean numbers which are going into our ML model. That's a substantial reduction in dimensionality. Is that what you were proposing?
Yeah, that's essentially it. I'm sure there are more sophisticated ways or different ways of doing it as well, apart from just mean. But the idea is the same, that instead of taking genes as individual features, you take the knowledge that some genes act together in certain pathways and then use those pathways as features.
Very interesting. I actually think that theoretically, a neural network should be able to identify these 15 groups as well without any external knowledge boosting. But doing so in small data sets of a few hundred samples, that I think is the main difficulty here. The loss functions of these complex ML models, they are usually non-convex, and their complexity only increases as you increase the dimensionality of your data.
I'm actually not sure about that. Like, I'm not sure that these pathway relationships can actually be extracted from simple sort of statistical relationships in biomolecular data, even if we had a lot of biomolecular data, because these relationships have required many different kinds of experiments over several decades. There are researchers who spend their entire lives studying these cellular pathways and networks. So I think domain, now that you've mentioned it, this domain knowledge integration will be crucial, even if we had thousands of patient samples, which we don't. But even if we did, I think this would still be relevant and really important.
Okay, all right. That does make a lot of sense. But wait, from where do you actually get this prior domain knowledge about pathways and various kinds of biological networks? Do you have some kind of a repository or a database from where one can freely download this information?
Yeah. There is a lot of this information is available out there for the public, and bioinformaticians and biologists are very familiar with them as well. Gene ontology is one of the most well-known examples that's used with biological data. There are databases for pathways like Reactome and KEGG pathway databases. There are databases about sort of protein structures and protein interactions like UniProt, String, Phosphosite Plus for protein modifications. And there are several mutation databases as well, like Cosmic and I'm sure many others. There is a lot out there. The ones I've listed, the list is not exhaustive, but certainly a good amount of knowledge is present in the ones that I just listed.
That's great. It will be very interesting to see, if you can actually incorporate this pathway and network information in your data and see if it helps in improving prediction accuracy. That will be really fantastic.
Yeah, I'm really excited to try this out in our project and to learn more about this research in general. Thank you so much, Kushal.
Thanks, Shefali.
Hi, everyone. I hope that, like me, you're now convinced that knowledge integration can be one of the ways of improving performance of your machine learning projects if you're using biomolecular data. In fact, if we think about AI applications more broadly in biological discovery and drug discovery, we should think about knowledge integration as an essential part of our data-centric AI toolkit. So before we close, let's take a look at three main ways in which knowledge integration has been described in literature. Now, one of the most common ways of adding domain knowledge in machine learning projects is by changing the input data. This can be done either through feature selection, for example, choosing which genes to include or exclude based on prior literature or molecular signatures or prior interaction networks, or a combination of several such methods. It can also be through feature transformation, like the example we discussed earlier, where instead of using individual genes as features, we can use pathways as features. A really beautiful example of this kind of domain knowledge integration is given in a paper called Network-Based Machine Learning in Colorectal and Bladder Organoid Models Predicts Anti-Cancer Drug Efficacy in Patients. So in this paper, the authors performed feature selection by first converting genes to pathway level features and then selecting only those pathways that were close to a drug target in a protein-protein interaction network. So if you think about it, the authors, in addition to using the biomolecular data, actually use domain knowledge in the form of pathways, in the form of protein-protein interaction networks, as well as prior knowledge about known drug targets. A second way in which domain knowledge can be integrated in machine learning models is through changing the model architecture itself. And this one applies to neural networks, where you can actually design your network in a biologically meaningful way. This idea is shown in a paper called Knowledge-Primed Neural Networks Enable Biologically Interpretable Deep Learning on Single-Cell Sequencing Data. In this paper, the authors designed a neural network architecture where each node corresponded to either a gene or a protein, and each edge or connection between the nodes was derived from a relationship or interaction that had been documented in biological databases. A third method of adding domain knowledge is in the form of some constraints during the model optimization process. A really great example of this approach is shown in a 2020 paper called Integration of Mechanistic Immunological Knowledge into a Machine Learning Pipeline Improves Predictions. And in this paper, features are prioritized or deprioritized during model optimization based on confidence in the relevance of those features by domain experts. Now, out of these three methods, the first one, which is changing the input data using prior biological knowledge, definitely seems to be the most common method of domain knowledge integration in machine learning projects that use biomolecular data. And as I mentioned earlier, I think we should really think of it as an essential part of a data-centric AI toolkit to systematically improve the data, to improve the final model performance. So please try out these ways of knowledge integration and let us know how they work for you.